All right, so I've shown you how we go from our genotype frequencies to our allele frequencies in the population, which is what we're interested in this case. And I've written those up here again for you. So the frequency of the big A allele, which we typically use the symbol P for, is equal to 0.56. And the frequency of the little a allele, which we use the symbol Q for, is 0.44, and those sum to 1. And now what we want to do is think about the formation of the next generation of the population. So if we're looking at the current generation, we've computed these frequencies, and now we want to think about forming the next generation. And remember, as geneticists, when we're thinking about forming the next generation, whether it's we're thinking about two parents and the offspring, or now that we're thinking about populations, we want to be thinking about the gametes, that is the sperm and the eggs. And so I'll draw a little symbol here for the male gametes and for the female gametes here. All right, and so let's think about now forming a random individual for the next generation. And to do that, we think about a process known as sampling the gene pool. So we randomly sample the gene pool in order to form a male gamete. And let's ask what would be the probability that that gamete would carry the big A allele. Well, of course, it is 0.56. So that's going to be P equals 0.56. And if that male gamete was to then fertilize a female gamete that was also sampled from the gene pool, and what would be the probability that it would carry the big A allele? Well, of course, that would be P equals 0.56. And so the probability that a child, let's draw the offspring over here, we'll keep them, the sex of them ambiguous. So the probability that that offspring is going to have a big A genotype is simply the product here equal to P squared. And that is, of course, 0.56 squared, which is equal to 0.3136. Now let's think about the little a allele. So if we randomly sample the gene pool to form a male gamete, what's the probability it's going to have a little a allele? Of course, that's going to be our allele frequency of Q, which is 0.44. And similarly, if we randomly sample and generate a female gamete, what's the probability it's going to have a little a allele? Well, that probability is going to be 0.44. And so when we think about the offspring, the probability that an offspring formed from the current generation is going to be homozygous for the little a allele is simply the product, which is Q squared, which is 0.44 squared, which is equal to 0.1936. All right. So those are the two cases in which we form a homozygote in the next generation. But we can also form heterozygotes by randomly sampling the gene pool, of course, by combining the big A allele with the little a allele. And so if we think about randomly sampling the gene pool and getting the big A allele, that's going to occur with probability 0.56. And if we generate a male gamete that has the big A allele, and then the probability that it will encounter a female gamete with the little a allele is equal to Q, so that is 0.44. And that's one way that we can generate a heterozygote in the next generation. But the other way is that if the male gamete carried the little a allele, which is going to occur with probability 0.44, and the female gamete carried the big A allele, which is going to occur with probability 0.56. So indeed, there are two ways to form a heterozygote. And therefore, when we do our computation here to figure out the probability of forming a heterozygote for the next generation, we sum those two together and get 2pq. And so the point here is that if we know the allele frequencies in the current generation, we can compute the expected genotype frequencies in the subsequent generation. And so, of course, those are going to be that the frequency of the big A genotype is going to occur with frequency p squared. The frequency of the little a genotype will occur with frequency q squared, and the frequency of the big A heterozygous genotype is going to occur with a probability of 2pq. And of course, these are all the possible genotypic combinations, and so if we added up those frequencies, they must equal 1. And so this is, in fact, the logic behind the following relationship, that if we add up all the frequencies, p squared plus 2pq plus q squared, it equals 1. And this is the famous formula known as the Hardy-Weinberg law. All right, and this is actually a very simple mathematical relationship, and you can see that this must be the case. If you recall some of your high school mathematics, we started off knowing that p plus q is equal to 1, and so it follows that p plus q squared must be equal to 1 squared, which of course is 1. And if you expand this, of course, you get this. And so this all makes sense.